Okay, so I'm going to hand over to Sam Reynolds now, who's going to tell us about the fascinating world of hoverfly migration. So over to you, Sam.
Okay, perfect. Well, hi everyone. My name is Sam Reynolds. I'm a PhD candidate at the University of Guelph, which is in Guelph, Ontario, Canada. And today I'm going to be talking about Syrphids more broadly, but most of the presentation is going to focus on a paper that I put out, I think about a year ago now, on hoverfly migration specifically, which is just a fascinating phenomenon. And you in the UK are a lot, quite ahead of the curve on hoverfly research, especially in regards to migration, which we will talk about today. Um, so a little bit about me, like I said, I'm a graduate student at the University of Guelph. So I'm in my fifth year of graduate school. I've been working on syrphid and native bee conservation and agriculture has been my primary area of focus. And this sort of hoverfly migration was a bit of a side quest for me. I became really interested in it. I had a couple people reach out and say that they had seen large amounts of hoverflies flying off of some bodies of water near where I was doing my research. And so I became quite interested in it. And when I went looking into it, I just found that the research was sort of all over the place. It was really hard to consolidate and to find. A lot of the papers were not digitized or in different languages. And so I thought, maybe part of my uh, to give myself a little break from IDing bugs, I would work on a review of hoverfly migration, and that's sort of what brings me here today. Um, this about me just kind of goes over my collaborators. So I work with Canadian Wildlife Federation and the Agriculture and Agri-Food Canada branch of the federal government in Canada. And I'm funded by a national funder called Unsung as well. So that's just a little bit of credentials, I guess. And yeah, I will jump into it. I don't know sort of the scope of the audience today, so I'm going to do a bit of a background on who are the syrphid flies. And basically, I think in the UK you would know them as hoverflies. In North America, we often call them flower flies, but also sometimes hoverflies as well. So they are a fly with many names. They're a family of true flies, so the order Diptera, and they're well known for their Batesian mimicry of bees and wasps. Um, and there's approximately 6,300 species worldwide from four major subfamilies. And Here is just an example of what I mean when I say they are, when I talk about their Batesian mimicry. They are often mistook for bees and wasps in pop culture, in magazines, in newspaper articles. So here are just some examples of people using photos of syrphids in order to talk about bees. And so that's kind of why I'm so passionate about talking about syrphids because I want to get the word out about them and help people to know how to tell the difference. A really cool thing about flies is that they don't sting. So they may look scary, but they're not. And one way that I can quickly tell if it is a syrphid or a bee is by looking at the antenna. So the antenna are a lot shorter for the syrphids. And they also only have two wings. So that diptera is diptera, meaning two wings, whereas the hymenoptera, so the bees and the wasps, are going to have four wings. They'll have four wings and hind wings. So that's just kind of a quick, easy way to tell if you're looking at a bee or if you're looking at a hoverfly. They're really impressive in their mimicry. So Batesian mimicry is a predator avoidance strategy where the harmless species or the mimic evolves to make themselves look like a dangerous species. So they make themselves look really tough and scary. And there's imperfect mimicry, and then there's also perfect mimicry. So these are examples of imperfect mimicry where they've sort of just evolved to have that black and yellow striped look that we're all kind of familiar with. Um, and then here's an example of perfect mimicry. So syrphids are really impressive because they can do really, really impressive things with their mimicry. So this is a bald-faced hornet on the right, and then on the left is Spilomia fusca, which is a species of hoverfly. And one of my favorite things about them is that they won't just mimic the appearance of bees and wasps, but they'll also do behavioral mimicry, which I find very funny. So like I said, one of the easiest ways to tell that it's a fly and not a bee or a wasp is those short antennae. And so what this species of Temnostoma has done is they have evolved dark forelegs. And those forelegs, they hold them up above their head to make it look like they have long antennae. And they'll even do it while they're flying. You can see on that bottom right photo, which I just think is really cute and very funny to watch. But they do a pretty good job of pretending that they are yellow jackets. But yeah, so I got really interested in hoverflies when I started my research. I wanted to do conservation of native pollinators. And so I kind of took on all of the families of native bees in Ontario. And then I also wanted to include sort of the second most important pollinating group for us, which are the hoverflies. And recently there has been an increased interest in syrphids and hoverflies. We had a field guide put out by actually one of my advisors, which is the field guide to the flower flies of northeastern North America. And I don't remember when that was published, but fairly recently. I think maybe right before I started my degree, which was in 2020. But yeah, so that kind of field guide helped to gain a lot of traction. I know in the UK, people have been interested in hoverflies for a long time and have known a lot. It's a lot less known in North America. And so with this guide, people have been able to sort of go out and it's really piqued interest. I included a graph here. This is just the number of publications on syrphid migration throughout the years from starting in 1840 to 2023. And you can see a huge uptick in the last, 15 to 20 years for sure. It's becoming a lot more of an interesting topic for people. And we're using hoverflies more in things like IPM, and they're being recognized as important pollinators as well more recently. So I think interest in these in this family will only grow. So I'm excited to share a bit more about them. In terms of their larval behavior, they're really interesting larvae, and they have a stunning diversity for a single family. So it's quite impressive. There's about 4 big subfamilies in within the Syrphidae family, and each one has sort of a different type of larval niche, which I think is really interesting. And they can roughly be divided by subfamily. So it's not perfect. There's obviously exceptions and things like that. But the 4 big subfamilies are Syrphidae is one, and they're Ephidophagous. So you can see one of that photo there. The larva is making quick work of eating an aphid, and they're actually pretty ferocious aphid feeders. Aristallinae is another subfamily. They're the most kind of diverse, and they tend to be the decomposers, living in sort of decaying organic material, detritus, things like that. Then the Pepizinae are also Ephidophagous. The Pepizinae subfamily, a lot of smaller flies, flower flies, so or hoverflies. Sorry, I'm trying to stick with the hoverflies today while I'm presenting. But yeah, they tend to be darker, smaller. And then Microdontinae is just a fascinating subfamily. They're ant specialists. The larvae look so different, and they tend to infiltrate ant colonies and feed on ant larvae and pupa. So yeah, all very different and very interesting. And then the adult behavior, it's mostly uniform adult behavior. So they tend to, they tend to eat nectar and pollen. They've got short, spongy mouth parts, and so they're much better equipped to eat from very shallow flowers. They tend to prefer like white, yellow kind of flowers and ones that are a bit more shallow for them so they can get their tongue in there. And what I'm here to talk about is that some species of adults are migratory. So this is a pretty bad video, but one that was taken in 2017 in California. And it is an example of someone who just kind of happened upon a hoverfly migration event, and they happened to be a dipterist. They happened to study flies, and so they recognized what it was and took this video. I'll show you if it will play. You can see the black dots flying by are just a couple different species of hoverflies racing by them. And this went on for quite a long time. So, we don't really know how many, but some were captured and identified. But yeah, that's the magic of the hoverfly migration. Um, so to get into some of the sort of evidence for migration, it's still not fully understood, especially in certain areas. Hoverfly migration is considered a partial migration, so meaning not every individual from a population is going to migrate. Some will stay and overwinter, and some will migrate. This is likely because of a strategic trade-off between avoiding competition and harsh winters and then avoiding the energy cost and risks associated with migration. And part of what I think that we need to focus on is understanding sort of what individuals are choosing to migrate and which aren't, and like for what reasons, because that's still not something that is really well understood. Most of our evidence for migration comes from Europe, like I mentioned. However, more research is starting to emerge from Asia, Australia, and North America as well. There's quite a knowledge gap in the global south, in South America and in Africa, which is likely attributed to many factors like funding, collaboration disparities, infrastructure deficit, things like that. So we mostly just make predictions and inferences for those areas right now. Not to say there's not research happening, but it's not in the published literature, so I wasn't able to access it. The graphs here kind of just show sort of the pink bars on the bottom graph are migratory species that are present. So species that are known to migrate elsewhere, but haven't been confirmed on that geographical region. And then there's also a blue bar for the species is present and we've confirmed it migrates through the literature. So you can kind of see that Europe is definitely dominating there. And then most of our evidence for migration has been observational, but there's kind of more experimental studies coming out more recently, which I think will be really helpful for us to understand sort of what's going on. So when I did my review, we looked over 50 papers. I think it was actually close to 60 papers, and 46 species of hoverflies were identified that had evidence for migration. If you want to see that list, if you're interested in the sort of the specifics, you can use the QR code on this slide to scan, and it should bring up a Google Doc with the full list of the Syrphid species, or you could also go to the paper that I mentioned, which I will, which is one of the references here, the first reference. But there are kind of 5 main species that are the focus of most of our research on migratory hoverflies. We have Episurphus balteatus, which is most of the papers mentions. Aristelis tenax is the second most, then Eupeotes corollae, Sceva pyrastri, and Syrphus ribesii. So those are our kind of big stars of migration, and we've done the most research on those groups and those species. So this slide basically just breaks down the migrants by subfamily, which I find interesting. So like I mentioned, they all have kind of interesting larval niches. You can see Aristallinae on the left here is also known as the rat-tailed maggot, which is not a very kind or generous name, but that's its common name. And those larvae tend to have a very diverse larval habitat. Most are in saprophagous environments living in decaying organic material. They're filter feeders, hence that little tail that they have. And then the Syrphidae is what the majority of our known migrants fall into the Syrphidae family. The majority of them are the aphid predators. Some are generalists, while others are specialize on just like a few aphid species. This sort of trend towards Syrphidae as the main migrant group might partially be because Syrphidae are much more well studied than Aristallinae, because they're the biological control agents. So because they're really useful in agriculture since they're eating those aphids, we tend to know more about them, and they're also tend to be in more human-dominated landscapes. But Syrphidae are a lot less diverse than Aristallinae, which suggests that migratory behavior is common throughout the whole subfamily and probably an important aspect of their biology. It's possible that they've evolved this migratory behavior to pursue and exploit migratory prey. So a number of aphid species are considered kind of migrants very loosely. They'll often kind of get taken on the wind and get sort of just moved. And so it's less of a sort of purposeful migration, and more of just kind of a dispersal. But aphid density is considered to trigger migration in some hoverflies. So it's likely that is part of the evolution of the Syrphidae subfamily. Aristallinae migration, on the other hand, could have evolved in response to other pressures, like the need to exploit ephemeral aqueous larval habitats, similar to what we see in dragonflies. But yeah, those are the two subfamilies that kind of make up our migratory hoverflies. And like I mentioned, most of the evidence for migration is observational. So this, in the literature anyway, dates back to 1818. There is a paper that was put out that basically describes sort of swarming and mass groups of hoverflies landing on ships out at sea, and it is kind of the first record of migration. We've also seen a lot of observation in European mountain ranges, which have been the most compelling evidence because we're seeing massive amounts of syrphids being funneled into small areas because of that geographic barrier. So the way that we've discovered insect migration and the way that we've been able to observe it is by looking at landscape barriers like oceans and mountain passes, which create this sort of funnel effect, especially in the mountain passes when there's sort of like a dip. It'll funnel all of the migrating hoverflies and kind of congregate them en masse to make it a lot easier to observe. But there's other forms of evidence as well, which I am going to go through today. That's kind of a big chunk of the paper, was looking at the different types of methodologies for studying this migration and seeing sort of what works and what doesn't, and how do we have the data that we have and where can we go from here. So the other forms of evidence are mark and recapture, vertical looking entomological radar, stable isotope, behavioral, morphological, physiological, and genetic evidence. So the first one being observational evidence. I think this is my wordiest slide, so forgive me. But like I mentioned, Europe has lots of studies. So this map is a map of Europe depicting locations hoverflies have been observed migrating. Most are located along coastal areas or through mountain passes, like I mentioned, and you can see there's quite a few red dots. And that's much more than what we would have in North America. In Europe, there was a 12-year observational study in the French Alps, which has really helped us to gain a lot of information. And also another observational study of, I think, 200,000 syrphids moving against the wind in Venturac River Valley, which kind of helped us to understand that this was like an intentional migratory event. In North America, there's not really been many observations. The earliest was in the early 1900s in New Jersey with Aristalis tenax, and then not again until 2017 with that video that I showed you on the west coast of California. In Asia, they've done multiple studies, including observations of migration in the Himalayan pass in Nepal, and they also conducted a 16-year observational study trapping hoverflies, which showed pretty strong evidence for migratory movement in the spring and in the autumn. And in terms of South America, Australia, and Africa, most of that evidence is inferential and not observational, and it's severely underrepresented in the literature. So that would be one area where I think that we definitely need more research energy going there. We've also done some mark and recapture studies for hoverflies, if you can believe it. I know they're very small compared to what we're used to seeing with mark and recapture. So we've seen some success using this technique on larger insects like monarchs in the photo. The problem is there's generally very low recapture rates and then a high reliance on community science, which for the large charismatic species can work well, and then a little bit less well for hoverflies. But in the context of flowerfly migration, there was a study done where Albert and Goldlin conducted an experiment in the 1980s using basically giant homemade tents, very big kind of malaise traps, where they captured some hoverflies at one location in a mountain pass. They marked them and then were recaptured 3 days later, even with significantly bad weather, and they were able to find some of their marked specimens that had traveled 111 kilometers southwest. So that was like a very insightful way to see sort of how fast are they traveling, even in bad condition. And so I think more of this coming out in the future would be really helpful, especially combining it with radio telemetry as soon as they can get the technology small enough to work on hoverflies. Right now, we've used radio telemetry with monarchs, the chestnut tiger butterfly, even with bumblebees, which bumblebees are probably the most comparable to some of the larger hoverflies. So I think we're kind of getting there on the technology side of it. But I think that would greatly improve our understanding of sort of where they're going and what they're doing. Another more kind of newer and exciting technology they've been using to study hoverfly migration is vertical looking entomological radar, which is a very complicated tool that I have not used myself, but is essentially using a beam system that allows them to kind of gather information on migrating insects in a little slice of the sky. And so it can give information like the number of insects flying, the size of the insects, the altitude they're at, and their wing beat frequency, which is all super useful. And there's been a series of papers in the UK coming out that just kind of shows like how useful this technology has been, and it's taught us so much in recent years. This is how we've been able to estimate that around 3,200 tons of insect biomass is crossing over southern UK each year during migration, which is just kind of a stunning volume and hard to picture. But within that, up to, I think, 4 billion is the estimated hoverflies of two species, Eupeodes carollae and Episurfus balteatus. So that's just two species out of 46 that have been monitored using this VLR technology, and that's 4 billion species or 4 billion specimens, sorry, traveled over the southern UK while they were migrating. And that kind of, we'll talk more about this paper that I'm referencing here, reference 15, I think. It's the Watton paper from 2019, and we'll talk a bit more about it when we get to sort of the ecological implications, but it's been a really, really useful tool for sure. I think the only kind of downside to this tech is it can't distinguish between species, so there still is some collection required, which is kind of technically difficult when they're in that high altitude of flight. But still, I'm really excited to see what kind of comes out in the next few years using this tech. The other thing that we've been using more in North America is stable isotopes. So this is kind of more in my wheelhouse to explain because I did, my background is in chemistry, but basically you're measuring the ratio of stable, so like the non-radioactive isotopes within a sample. Usually you're using like a mass spec machine, and this provides information about the origin of the fly and its diet and environment. So kind of the principle behind this technique is that organisms incorporate an isotopic signature from their food and environment, which becomes sort of embedded in their tissue and reflects the history of where they've been, where they grew up, what they were eating as larvae. And that is usually kept in sort of the wings and legs. And so you can use that to understand where the larvae were originated. So if you're, doing stable isotopes on a suspected migrant somewhere, and then you see that it's from, somewhere 3,000 kilometers away, then that gives us some important details. The only, I guess, problem with this technique is that it's a lot less precise, but you can make it more precise by adding different elements into it. So you can add not just hydrogen, but also carbon, oxygen, strontium, and then metal contaminants are really helpful too in narrowing down sort of a more specific area. This is a really fast growing field. We've seen it with dragonflies, butterflies, moths, and flower flies, hoverflies. In North America, my colleague on this review, actually, Dr. Scott Clem, he was able to perform stable isotope analysis on Eupeodes americanus and actually determined that they had traveled 3,000 kilometers, which is just kind of mind-boggling for these tiny creatures, and is also performed in France, and 1,400 kilometers is what they were able to determine that the flies had traveled. So I think this is also a really exciting sort of technology to help us to understand sort of how far these little flies are traveling. There's also been quite a few behavioral studies in which hoverflies are restrained and manipulated to observe the timing of flights, how many initiations of flights there are, and direction of flight, which has taught us a lot already about hoverfly migration. So these can be lab or field experiments, often using a tethered flight mill, which I've pictured here. One study was done that was able to determine the heritability of migration in flower flies. So basically, they examined flight behavior of field collected migrants, field collected resident phenotypes or non-migrants, and then they lab reared their offspring, and they found that the offspring of migrants initiated significantly more flights than the offspring of overwintering hoverflies. So that was a really interesting study, and it was sort of the first of its kind to show that there's potentially a heritability component of this. They've also tested their navigation ability using these methods, and basically they've shown strong biases for hoverflies orienting themselves towards the south in autumn and then towards the north in spring. I think it was that 93% of the time they flew south in the autumn and 73% of the time they flew northeast in the spring, even in sort of varying weather conditions. And they also used these behavioral studies to determine that flower flies are likely using a time compensated sun compass to navigate, which is what we found for a lot of other migratory species. So yeah, these are also very useful studies for sure, and hoping to see more like them coming out. And then last, I think no, there's a couple more. So then morphological studies is another area of research for hoverfly migration. Migration is known to affect wing and body shape and size in migratory insects. So again, in butterflies, dragonflies, we've seen kind of adaptations towards their body size and wing size. Some evidence in hoverflies has shown that migratory females are larger than their non-migratory counterparts, which has been like an indicator that there are some morphological changes happening. And then another study by Dr. Clem showed that Eupeodes americanus migrants have larger body mass and greater wing size compared to non-migrants. I think there's been a couple other studies that kind of have shown mixed results or kind of inconclusive results. So we're pretty new on this kind of stuff right now. Like we don't know for sure that they have very clear morphological changes. Sorry, I realized I hadn't had a glass of water in a while. And then physiological studies is kind of a little bit similar, but evaluating sort of the physiological data to help determine which individuals are migrants in a population. So like I mentioned, there is that sort of not all of the individuals in a population will migrate. And so being able to determine which are more likely to do that, and if there's a way to tell, or even to tell if a species is migratory, are these kind of physiological studies. So some of these are upper and lower temperature thresholds, and then using photoperiod as well. So I think there's a couple studies here, a couple papers that I referenced, but one study looked at Episurface balteatus compared to Syrphis rubesii. And what they found was that Episurface balteatus was not highly cold tolerant, so much more likely to a high proportion of its population would migrate in a given year, compared to Syrphis rubesii, who seems to be able to have a higher tolerance and be able to survive overwintering. And then they've done some photoperiod studies as well to understand what kind of induces migration. The photoperiod is just kind of the duration of the light in a 24-hour cycle, which varies with the seasons. And they found that reproductive diapause could be induced in Episurface balteatus at a critical photoperiod. And that critical photoperiod was basically similar to what it would be like in September in Germany. And that's kind of what they were studying was at what point it is migration induced in these species. And then the last in terms of evidence that we have, which I hope I'm making a compelling case that we know lots about hoverfly migration, but still so much to learn. The last evidence is sort of genetic. So I've split this up into transcriptomics and then population genomics and metabarcoding. In terms of the transcriptomics, this kind of just examines the RNA expression. This was done in I think in 2022, a paper came out that described a draft genome and compared migrant and non-migrants of Episurface balteatus, that really common sort of model of a migrant. And And they found 1,500 genes that were associated with migratory behavior. Most of those were things like fur metabolism, muscle structure, hormones, flight, feeding behavior, things like that. And I think it'd be really interesting to link those genes with the migratory polymorphisms that we've talked about in the morphology, to see, are we able to link these genes to the actual physical changes that we're seeing in the migratory individuals. And then the population genomics and metabarcoding has also been really interesting. In Europe and China, several studies have shown high levels of gene flow between geographically distant populations. So, you wouldn't expect to have this like genetic admixture between two very separate, distinct populations, but because migration is likely occurring, they're seeing that there has been gene flow between the populations. And that's one kind of type of evidence for the migration, even if we haven't monitored it directly. And then a really cool paper that also used pollen metabarcoding has been shown to reveal flight paths over long distances. So that was something I kind of wouldn't have thought to do, but I think has been really useful. Basically, taking the pollen off of the specimens once they've been collected to determine what they've been eating. And this study actually found spring and summer migrants consistently had pollen from a specific citrus species found only in central and southern China, while the autumn migrants had pollen from chrysanthemum, which was restricted to northeastern China. And so not only did this work kind of reveal where they were coming from and what they were eating, but also, like what plant species were a crucial food resource for them. And I think that can be really helpful for knowing which plant species to kind of conserve and could be a special interest for hoverfly conservation policies. This is a graphic from our publication that kind of just depicts the current known migratory pathways. So this is basically all the research that we have so far of where the migration is occurring and what pathways they're using. So this is not to say this is all the migratory pathways that happen. I'm sure there are many, many more, but this is all we have evidence for right now. And I'll leave it up for just a couple seconds. You can look. And then I also put up just kind of common migrants in each of the geographic regions as well. But yeah, so that gets us to sort of the part that I'm most interested in, which is the ecological impact of this migration. So you've heard lots of evidence of and lots of methodologies, which some people are really interested in the methodologies. I'm like most interested, I would say, in sort of the why behind why do we care about hoverfly migration. I mean, there's tons of reasons too, but one is that they're really important pollinators. So they're pretty great pollinators of many crops, things like canola, sweet peppers, strawberries, especially here in North America. I think the research says that they pollinate about a third of our canola, which is a huge export for Ontario. So they're pretty economically significant. And they're also generally considered the second most important pollinating group after bees. They fill a different niche. They're able to pollinate at different times of day and be a little bit more resilient to things. So they fill a different gap than bees do in our pollination. But the interesting thing about pollination and migration is that they are also doing this pollen redistribution and potential gene flow across large distances for plants, which can increase the genetic vigor of plants. So that paper that I had referenced earlier, the Watton paper, showed that billions of pollen grains are transported per year during flowerfly migration, which is just huge. And this is contributing to, cross-pollination between Britain and continental Europe and probably plenty of other places, but this is what we have evidence for. And we also think that they have relatively high floral constancy based on our current data. So for those that don't know, floral constancy basically just means that they're kind of going to the same species of flowers throughout their trips, which is very useful when you're thinking about pollination. Obviously, you want them to bring the pollen from one species to that same species of plant in order to actually have pollination occur. So when I first read this, I was like, well, how do I know that the pollen is actually viable? How long do they last? So I did look into that, and it's really highly dependent on the plant species and the number of environmental factors. But there was a study on canola, which is a hoverfly pollinated crop, that suggests a pollen viability of up to 34 hours, giving hoverflies time to travel hundreds of kilometers before depositing viable pollen. So I do think that this is a pretty interesting thing that we should be looking into, because it's probably a very helpful thing that these insects are doing that we just haven't really spent much time thinking about. We also, in terms of floral constancy, there is a paper that showed that males had an average of 1,100 pollen grains on them when they were caught, primarily of one group of flowers, rubus pollen, in their gut analysis, while females had an average of 210, primarily of senecio pollen. And so that's showing that there is, likely the bias could be based on availability, but there's, I think, one other study that has also shown something similar, that they tend to have a very limited amount of flower pollen at a time from a limited amount of species. So that does imply that there's a substantial degree of floral constancy in these species of hoverflies. So probably they are doing some pretty important pollination along their migratory journey. And this is just kind of an example of sort of what I'm talking about here, where they'll collect the pollen from, this is a canola flower, and then travel and deposit it on another canola flower, hopefully a large distance away. And then you can also see, I've depicted a very rudimentary larva eating aphid, showing that once they get there and then they lay their eggs, which is most often what happens, the female will lay their eggs once they arrive, then you're also contributing to that biocontrol. And that brings me to kind of the second large ecological impact, which is the biological control. And this is also the most economically impactful part of hoverfly migration. So Episyrphus balteatus alone, if we're using that as our kind of model migrant, they will eat up to 400 aphids, around 400 aphids during their development as larvae, and a single adult female will lay around 400 eggs. So if you do the math on just kind of one female arriving after migrating, that's like 160,000 aphids being eaten because one flower fly migrated, and we know lots of flower flies are migrating. So it actually puts a pretty big dent in those aphid populations. One study showed that approximately 1 million cereal aphids per hectare of cropland in Britain is controlled by offspring of migrants each year. So definitely not insignificant. And there's also been some long-term studies in Germany that have linked fluctuations in the aphid populations to the timing of Episyrphus balteatus presence in the springtime. And so I do think those two things are likely linked and require a bit more research to kind of fully parse that out. But yeah, it's definitely an important aspect of their biology. And the amount of biocontrol services that they're contributing is probably going to vary significantly depending on lots of factors, but is really important for our agriculture. And then the last ecological impact that I'm going to talk about is nutrient transport. So they're not just transporting pollination services and biological control with them, but it's also estimated that Episyrphus balteatus and Eupeodes caroלי transport up to 2,500 kilograms of nitrogen, 250 kilograms of phosphorus, and 150 gigajoules of energy per year during migration. And that's just like, a really large amount of nutrient being transported. There's also lots of studies on things like periodical cicada emergences and locust swarms that show really useful resource pulses just from, bringing in a food source for birds and other animals. And that's likely happening as well, but we don't really have any evidence yet in the literature for that. But and the last thing is it's also possible that hoverflies are carrying pathogens across long distances, but that's basically not studied at all. So I just wanted to say it's not all good, I guess. We just have a lot more to learn about that. And then I included a slide here about hoverflies and climate change because I've mentioned how important economically and ecologically that these flies are. And one thing we have to consider is that the aspects of climate change on insect migration is just going to be a critical area of future research. We've seen that storms and changes in wind directions are likely the greatest contributors to migration mortality. There's one paper um, that found tens of thousands of migrating hoverflies dead on strand lines and beaches in 2022 because of a storm that came in. Evidence shows that fluctuating monsoon seasons in East Asia can shift the migration of brown plant hoppers. And so that's probably happening for other species as well, and we just don't know. Um, warming temperatures, changing wind patterns, all of that can significantly alter migratory pathways and even the migration triggers. And to know if those are being altered, we have to first understand what they are. And that's sort of the stage that we're in now, is that understanding of, what are the triggers, and if we're relying on these flower flies and the hoverflies to come in to agricultural areas and control aphids, but they're delayed, or those are things that we need to know and consider. So that's, I think, going to be an important aspect in future sort of migration work. And that kind of brings us to future directions. So in terms of research, I think our future directions in this field are going to be looking at mountain ranges like the Caucasus in the Middle East or the Himalayas in Asia. Unfortunately, in North America, our geographic barriers aren't oriented correctly, and so they're not probably going to be acting like funnels. So we're going to have to find other innovative ways to study our migration. But I think in these other areas, they could be important to monitor. And also just monitoring where we know other migrations are happening. So oftentimes the way we've discovered hoverfly migration is that there's people studying Lepidoptera, people looking for butterfly migrations. And then they're just like, hey, that's weird that there's a bunch of flies here too. So I think kind of being very mindful that, reaching out to researchers who are doing migratory work on other insects, and seeing if they can also keep an eye out for hoverflies and let us know if things are happening so that we can go set up and monitor there is going to be important. We're also going to see a lot of advances in methodologies and technologies. So like I mentioned, extremely lightweight GPS telemetry will be really useful. Advances in our modeling and our statistics, and the genomic techniques as well is going to be huge. And then, trying to combine stable isotopes with things like morphological and physiological data will help us to answer important questions like, where are they migrating? Why are they migrating? How are they migrating? Lots of things that we just still don't quite know. And then for future directions in conservation, I think more research and clear conservation strategies are imperative. We might lose some of these fascinating species before we truly understand them. And I think that would be a real shame. Native pollinators we know are declining globally. Evidence of hoverfly declines, however, are really conflicting. And most of it evidence is only coming out of Europe. So we don't really know what's happening to them in other places. Some studies show that they are not declining, that they're going up, but that they're shifting their range. But those range shifts are often to places that they're not going to be as agriculturally important. And so it would still have a huge impact on us. The migratory individuals might be more resilient to impacts like habitat fragmentation and deterioration because they're able to disperse. And so we definitely need more research there. And just in terms of like general long-term population monitoring is needed, because it's hard to say if a species is in decline if we don't know what its baseline is. And so having more of that monitoring is going to be really important. And then lastly, providing semi-natural habitats for both adults and larvae so that we can support hoverfly populations throughout its entire life cycle as well. For my conclusions, we need more field and field realistic experiments to understand how the natural environment affects these hoverflies in their flight, the length of their flight path, lots of things about their migration that we still need to understand. We need much more published research in South America and Sub-Saharan Africa. And we need to prioritize hoverfly conservation because while they're little and not often as well known as some of the more charismatic species, their impact is still really important. So that is all I have for you today. These are my acknowledgments. There's lots of people to thank, but especially Dr. Scott Clem, who I worked on this publication with, and then everyone who helped me track down every piece of literature that there was on hoverfly migration, my lab mates, and especially the hoverflies that have died for this research. I always like to acknowledge them as well. So yeah, feel free to reach out to me with any questions, or we can take questions now. But thanks.